Okay, you can see, okay. Yeah. Hi, I am Gerard Go, and I'm a computational protein virologist. I basically study proteins. I look at behavior of proteins and examine if there's any unusual activity going in protein, and then try to figure out what is going on. So I have been researching SARS-CoV, HIV, a long list of viruses since 2005 when I finished my work at Indiana University School of Medicine. Now, as you know, that the SARS-CoV started in December. The focus of this, the initial outbreak was at the Wuhan market. Now we know that it probably did not arise from the Wuhan market, but we know there is some kind of animal intermediate in there. Then later on, people begin to think, where did this come from? Where did the virus come from? Was it man-made? And some people suggested that maybe China created the virus and sort of thing. And there was a very important paper that came out by, this is a landmark paper, by Anderson of Scripps Institute, and hold a big group of researchers that pointed out that this is a this virus has a natural origin because it's so novel, especially when you look at the spike protein. The spike protein is a surface protein that binds to the human ACE receptor. And they suggested, and is so well adapted to the human ACE receptor that many scientists, including the group, believe that that this virus had been in human for a long period of time. It didn't just occur at the Wuhan market. The question is, how the heck did it remain in human for so many years without the notice of doctors? Now, this research I've been done points out that pangolin allowed a vaccine strain many years ago, and it spread. There was a silent spread for a long period of time. Now, the important tool that we are using right now, an important concept we are using right now, is something known as protein intrinsic disorder. It basically means that all it is that parts or portions of a protein that has no structure at all. We use tools like artificial intelligence, database design, statistical technique, molecular graphics, and modeling of proteins. There is, the thing about structural biology. Structural biology is the main theme in biochemistry, whereby the structure of the protein defines the function. But there is an open secret that many proteins do not have structure at all. Now, this is called unstructured, flexible, unfolded, intrinsically disordered protein. Now, this is the main focus of our study. Now, here's an example of what is structured and unstructured. We use AI to scan through the sequence And then the rate, this is the SARS-CoV protein, which is one of the inner, which is an inner shell protein of the SARS-CoV-2. The red one is disordered, and the blue one is ordered. So the red one is moving. Now, so the other thing that we use, another tool, is something called a protein predictor. The particular predictor we use is something known as a VLXT. It was first created under the leadership of Keith Dunker, my ex-boss at Washington State University, and later the rector at Indiana University School of Medicine. You will see PONDR VLXT over and over again. This is the neural network, the way an artificial intelligence tool, the input in this case, it's a sequence of the proteins, the hidden layer, and the output. The output is order or disorder. You can train the neural network based on what is known about the protein. Here it is. The input is a sequence, the PONDR neural network, order, disorder, the database of order and disorder, the output is order or disorder, and the information is placed in the database. Now, another ratio we will be using again and again is something called the PID, or percentage of intrinsic disorder. This is basically the number of disorder divided the number of the total number of residue. The higher the the PID range between 0% and 100%. The higher the PID is, the more disordered a protein or protein chain is. Here is the coronavirus. The outside, the external is made out of surface glycoproteins, which includes the S protein, and the middle one is the membrane protein. Then you have the N, or nucleocapside protein, and then right in there is the RNA. We'll come back again and again. Just remember that the M protein is the membrane protein. The outer shell plays an important role in protecting the virus from damage. The inner shell, the M protein, help to facilitate the rapid replication of the virus. And there's something very, very strange I see in the SARS-CoV from the very beginning. The M, this here, is extremely hard, harder than most of its family, the coronavirus family. Very, very strange. There is something very, very strange about this virus. And before the 2003 SARS-CoV, coronavirus was not very interesting because it caused mild cold, and that's all, with very low rate. It became interesting, of medical interest, only in the 2002 and 2003 outbreak. Before in 2005, when I came back from America, I was interested in studying viruses, especially the shell of the viruses. I started off studying apparent I launched a parent project of this particular project to study the shell of the HIV. And I found very strange thing about the HIV. The outer shell of the HIV is extremely disordered. It's moving like crazy. That is why it was extremely difficult to find a vaccine for HIV. For 40 years, people have been looking for a vaccine for HIV to no avail. And up to today, there's not a single HIV vaccine. Then there was an offshoot of this project. One of the offshoot in this project that I started before the 2012 MERS outbreak. And this was, I started studying the disorder level of the M and the N protein. The M, like in monkey, is the membrane protein. There is the outer shell of this thing and it's protecting. And I found, then I measured the M protein, the N in the nucleocapside protein, and a model came out of it. Here again is the M protein, the N protein, and the RNA. Very after studying what was known about the behavior of animals. Remember that before 2002, we didn't have a lot of knowledge about human coronavirus because they were not interesting, but we did have a lot of knowledge of animal farm behaviors, especially porcine, that is pig coronavirus. And when I measured the N and M, it broke up into three groups. This was actually before the MERS outbreak in 2012. And I had published this, and I published the paper out in 2012 that the coronaviruses in general broke up into three groups. Now, the A groups are those with high respiratory transmission level and low fecal-oral level. We know from animal studies that coronaviruses have levels of fecal-oral or respiratory this thing. By measuring especially the N protein, the N disorder, NPID, the higher level NPID tend to cluster with higher levels of respiratory and lower level of fecal-oral. Then there's a whole bunch of intermediate. SARS came in to the middle, and we didn't know about MERS at that time. Then MERS came in. SARS-CoV, the 2002-2003 SARS, fall right in the middle. Intermediate level of respiratory and intermediate level of fecal-oral transmission. As you can see, that SARS has a 50.2%, 50.2% and 8.6. If you look at the N value, NPID value, it fits right in the middle. Then the MERS came in 2012. It was an opportunity to validate our model. Now MERS came out very interesting. It falls into group C. The category that has lower level of respiratory transmission. Oops, how do I get? lower level of a level of respiratory transmission, but a higher level of fecal-oral. And that, bingo, that actually validates our model, because MERS is very difficult to transmit among humans, unlike SARS at that time. MERS originate intermediary are camels, and camels, they move very rapidly by fecal or urine. It, it seems that MERS, there are little quantities of viral copies, viral particles in the fecal material of humans or camel, but in the urine there are large quantities of viral particles. So that validates. Then only 2020, SARS-CoV came in. This time, this is another chance for us to validate our model. And SARS-CoV again fell into group B, an intermediate level of respiratory and of respiratory and fecal oral transmission. Bingo again, bingo. It validates our model, because if it had fallen in C, it would have made this model completely nonsensical. We have to throw the model into garbage. But because it hits B, it validates our model. But then the thing is, it may sound contradictory. If SARS-CoV is more contagious than SARS-CoV, why didn't it fall into group A? Now, but then the thing is that in group A, it includes CoV of lower levels of fecal oral transmission. But as you will see later, SARS-CoV, that if it had fallen into group A, it would contradict our model again, but it did not. But as you can see, there's something else very strange about SARS-CoV. SARS-CoV has one of the hardest shell among its family. Why? We didn't know. I had no idea why it is like that. I begin to suspect that the reason why SARS-CoV was so contagious, because it is because of its hard shell. Remember, the hard shell, outer shell of the virus protects it from the environment. And in our mucus and in our saliva, there are antimicrobial enzyme to attack the virus. And because it's able to resist the enzyme in mucus and saliva, a large quantity of viral particle is expunged from the human body. And bingo, there are clinical evidence showing that SARS-CoV sheds a lot of materials into the saliva and the mucus. There are clinical evidence showing that. And then the thing is that why SARS-CoV? I didn't know at first. I have 5 papers on SARS-CoV out. If you Google Gerard, go Pangolin, SARS-CoV, you can find all my papers there. And it's only in June I did a study of I made a study, a search throughout this family, and I found that only burrowing animals and rabbits and pangolins have very hard shell. And because why? Why? Because these burrowing animals are in contact with buried feces that remaining in the ground for months and maybe even years. And by the way, rabbits CoV is not related to SARS-CoV. It's not closely related, but pangolin CoV is. Here it goes. Here it goes. You see that SARS-CoV is one of this is only a sample of the family of CoV, and SARS-CoV has one of the lowest in its family. And I didn't put pangolins. I put rabbit. And then there was another spin-off, another spin-off in 2015. I found that the inner shell of many viruses, including Ebola, flaviviruses, including dengue, yellow fever, and whole bunch of them, and the Nipah virus are correlated with the virulence. As you can see here, this is the inner shell, the gray is the inner shell. And why? As you see that the inner shell of SARS-CoV is correlated with transmission, respiratory transmission. But on the other hand, many viruses have inner shell correlation with virulence. Why is that? We'll talk about it later. Well, the reason is this. The reason is this. The reason why virulence is connected to the inner shell disorder, because the inner shell plays a role in replicating rapidly. And many of the viruses, they have something called a Trojan horse immune evasion. They replicate very, very fast before the immune system notices it. As a result, what happened, the more disorder it is, the more efficient the virus replicates. And by the same token, for a respiratory virus to be viable, to spread from one person to another, there has to be at least a minimal amount of viral load in the mucus before it spreads. So that is why there is correlation between all three. Now here is some of the data for the pangolin. Pangolin have huge. This is the three sets of pangolin in this is the N protein. Now the there is 4 sets. The 2019 one is very similar to the SARS and SARS-CoV, but the 2018 and 2017 falls in a different category. It's remarkable, because you know why? You know why that pangolin, even though pangolins and SARS-CoV, they share about 90% sequence similarity. It's very closely related. And you know why? Because pangolins have a lot of fecal-oral component in it. If the way they eat is pangolins are ant eaters. In they burrow in the soil to live in the soil, and they dig up soil and they flick their tongue to eat ants. Their sticky tongues in there. They don't have teeth, but they flick their tongue. And in the process of doing, they can accidentally eat fecal materials. That is why uh, the 2018 is has extremely low NPAD. That means, according to our model, our two models, one is the virulent model, and one is the CoV shell model. It is the 2018 has a slow spread, and not only so slow spread, it's an attenuated virus because our virulent model tells us that once it enters the body, the 2018 enters the body, it will spread really slowly, and that is why that is why uh, that is why it the the pangolin-CoV must have entered the human at 2018 or earlier. And not only did it enter human, it entered as an attenuated strain. Here is it again, the pangolin. The pangolin is, you can see that the big difference in the NPID. And then there is also there's also bad and the bad this thing, then the red G13 has 96%. So the mechanism, as I said, the, the outer cells allow penetration, but the disorder at the inner shell helps faster replication. And the reason is protein promiscuity, and inner shell is involved in the replication process. That is why the inner shell disorder is related to the virulence, both the virulence and the mode of transmission, particularly the respiratory transmission. There you see again, the pangolin is very closely related to the SARS-CoV. Now, okay, there is, what does it mean? What does it all mean? As for how do we compare the SARS-CoV 2002-2003 to the SARS-CoV-2? In the civic case of the civic cat, I looked all over, I wasn't able to find any great difference in NPID between the civic version of civic CoV and the human CoV. That means that it must have moved suddenly. There was only a short time span before it moved into humans, and it must have moved into humans as a virulent strain. That's why in 2002, doctors were able to discover the virus really quickly, and it suddenly disappeared perhaps because of restrictive orders that the government has placed. But then when you come to SARS-CoV, something else very strange is happening. The pangolin strain, the inner shell of the pangolin strain is very different. It's quite different from the human CoV. look at how low it is. Look at how low it is, 44 to 46%, while the human SARS-CoV-2 is 48%. That means that when when the virus entered from bat to pangolin, there was an evolution. It took many years of evolution before the pangolins was able to enter humans. The pangolins, because of its fecal-oral transmission behavior, allow the CoV, pangolin CoV, to adapt to pangolins. And when it transmitted to human, it entered as an attenuated strain with low virulent and low infectivity because it is most effectively transmitted to fecal and oral transmission. And that could have happened for many years before it became virulent and very highly contagious. That's why that protein, that could have been the reason why scientists all over the world have studied the proteins of SARS-CoV and found it to be very adapted to the human ACE2 receptor. Here it is. Our model has detected a vaccine strain of SARS-CoV entering the humans years ago and later became virulent. By the way, this is not the first time that a vaccine strain became virulent. We had this experience with the polio vaccine. The polio vaccine are made out of two types of vaccine, are mainly made out of two types of vaccine. There's a Salk vaccine that is made up of proteins, inactivated virus, and then there is the Sabin virus, the Sabin vaccine. The Sabin vaccine is an attenuated virus strain. Now, the Salk vaccine doesn't give protection for only a short time. Several booster shots is needed before it provides immunity for a short period of time. But the Sabin strain, which is an attenuated virus, one shot would give a lifetime of protection. But here's the trick. There's a price to pay for that protection. After a few years of distributing the Sabin vaccine, they found that the Sabin vaccine mutates back to a virulent cell. And this could have happened, this is likely to have happened with the SARS-CoV via Pangolin-CoV. Here is this, the mechanism. The inner shell protein assists the replication by recruiting other. There are important mechanisms. That is why the inner shell of a lot of virus assist in the replication, quick replication. And because in order to transmit from respiratory transmission requires a certain level, minimal level of viral load. The mechanism is the same as virulence. The difference between virulence and respiratory transmission is this. Virulence is viral load, where the viral load in important organs overwhelms the person. But the viral the respiratory transmission emphasize the viral load at the mucus or the saliva. So what happens in the Trojan horse immune system, sometimes this strategy backfires on the virus by killing the host because it replicates so fast in vital organs. Here it is, the immune system. This is what we have saying again. The difference is between them is a viral load in the vital organs versus the body fluid. So so a precursor SARS may have entered in as a mild bacteria in 2018. Not this is wrong. 2018 or below, and pangolins may explain many of the mystery, and that's also why SARS-CoV is so adapted to humans, even more so than the 2003. Actually, it also suggests novel strategies to develop 2019, better understanding of the evolution. Also, the other thing you have to there is also a mystery there. Look at China. China has 1 billion people. US has about 200,000 people, 300,000. Look at the number of infection of people in China and look at the number of death and infection in first people in US. There must, this epidemiological data suggests that somehow an attenuated strain must have spread slowly, silently for years within China. That is what I believe is so. And there is also, if you Google under my name, Gerard Go, smallpox, poliovirus, Ebola, flavivirus, HIV, you find papers in it. This pangolin papers had been published in Journal of Proteome Research. Is not freely available. You have to pay a fee, but I placed a preprint in my research gate. Just Google and you will find it. And this is my ex-boss, my thesis advisor, my ex-co-worker at Indiana University School of Medicine. And I have a book out. It doesn't cover SARS-CoV-2. In 2000. It was published in 2017. But you have plenty of stories about the SARS-CoV-2 and polio and HIV. You can get it in Amazon. And you can also visit my webpage about me, Gerard Go, and you can get a link to my papers, my research gate papers.